Hello friends, in the last video we discussed about natural selection. You can refer my video, the link is given in the description box. And today we are going to discuss about evidences of natural selection or role of natural selection. First is Lederberg's replica plating experiment. It was conducted by Joshua Lederberg and Esther Lederberg in E. coli, which is bacteria. Bacterial cells were first cultured on agar plate. And on these agar plate, many colonies developed. Then, velvet block was pressed against the surface of culture. Cells from each colonies were picked by velvet block. This block was then pressed against plate containing penicillin. Most cells failed to grow on penicillin medium, but few were able to grow. So what it was concluded that these cells which are able to grow on penicillin medium are resistant to penicillin. But how? Drug penicillin itself does not induce resistance. It was proved by taking cells from the original colonies that were never exposed to penicillin. Then these were tested for resistance to penicillin and it was found that these are resistant. So penicillin simply selects against susceptible bacteria by killing them. Or in other words, penicillin selects the resistant bacteria and kills those that are susceptible or sensitive. There are certain genes that are responsible for the resistance, and these develop due to the earlier random mutation. These are already present in the population. Earlier, these are present in the low frequency because these do not have advantage over the one which consists of the normal genes and are susceptible to penicillin because these were grown on the normal agar medium. But when these are grown on the penicillin medium, then these have the advantage over the normal ones. Hence, those which possess these mutated genes are pre-adapted to survive penicillin treatment. That is, these are already adapted to survive the penicillin treatment. It is not that these develop the resistance due to the exposure to the penicillin. In the presence of the penicillin, these reproduce and perpetuate. Therefore, next generation shows marked resistance to penicillin because in the next generation there are more resistant bacteria because these have the advantage over the susceptible ones. And continued selection, or when these are continuously grown on the penicillin medium generation after generation, gradually increased resistance. That is, there is gradual increase in the resistant bacteria in the population. Therefore, what we can say that natural selection has selected and supported the progeny carrying the beneficial mutation. And this results in the resistant bacteria to evolve, or hence evolve resistant bacterial population. Let us understand this Lederberg's replica plating experiment with this figure. This is the master plate, that is the agar containing medium on which the bacterial cells, that is E. coli, is grown. Then the sterile velvet is pressed against this. It gives imprints of all colonies. Then this is pressed against the two fresh media, one which lacks penicillin and other which contains penicillin. Then these are incubated. So in this medium all the colonies grow. Here only two colonies are growing. These are the penicillin resistance colonies which are able to grow in the presence of the penicillin. Others are not grown if we compare it to the master plate. So we can say that the other colonies which do not grow on this penicillin containing plate are actually the penicillin sensitive cells. So if we compare these two plates with this master plate, we can tell these three colonies. These are the penicillin sensitive and these are penicillin resistance. These are already present. It is not that penicillin's resistance develop when it is exposed to penicillin. It is only that natural selection, in this case penicillin, selects the penicillin resistant cells over the penicillin sensitive ones. Now the second is sickle cell anemia with relation to malaria. Sickle cell anemia we have already discussed in genetics. You can refer my video, the link is given in the description box. This sickle cell anemia is caused by recessive gene. And this gene is responsible for the production of abnormal hemoglobin. And the red blood cells which have this type of the hemoglobin are distorted or sickle shaped. Hence the name sickle cell anemia. Homozygous individuals which have 2 copies of this recessive gene die early in life due to severe anemia. But heterozygous individual which has only 1 copy of the gene have mild anemia. Also, these have one advantage. These are naturally protected against contracting malaria. But how? Because these heterozygous individuals have both normal and abnormal RBC. Parasite cannot live in these abnormal RBC which is distorted or sickle shaped, and so they are protected against malaria. Hence heterozygotes survive better in malaria endemic region. Endemic means disease or condition that is regularly found among particular people or in certain area. In these regions, population have both normal and heterozygous individuals. These heterozygous individuals act as carriers of gene for sickle cell anemia from generation to generation, and these are recessive gene which are responsible for sickle cell anemia. It is entirely disadvantageous in one environment and advantageous or beneficial in another environment. That is the environment where malaria is endemic. Hence can be selected in these regions or environment. This explains high proportion of this gene in geographical areas where malaria is endemic. Now third is malaria in relation to glucose 6-phosphate dehydrogenase deficiency. Glucose 6-phosphate dehydrogenase is an enzyme and its deficiency is X-linked hereditary disease. In glucose metabolism, conversion of reduced NADPH to oxidized NADP is catalyzed by glucose 6-phosphate dehydrogenase enzyme. Also, glucose 6-phosphate dehydrogenase deficiency is characterized by low levels of reduced glutathione in RBCs. Reduced glutathione level is maintained by glutathione reductase and its coenzyme NADPH. And we have seen earlier that this glucose 6-phosphate dehydrogenase is required for the adequate supply of NADPH. And this enzyme is encoded by the gene that is present in the X chromosome, which is sex chromosome. Glucose 6-phosphate dehydrogenase deficiency leads to hemolytic anemia and prolonged neonatal jaundice. Hence, if there is hemolysis of RBCs, therefore malarial parasites are unable to multiply in these RBCs because malarial parasite breed on these RBCs. Therefore, decreased glucose 6-phosphate dehydrogenase activity within their RBCs leads to resistance to malarial fever. This is beneficial in malaria infested regions and is favored by natural selection. Example, 50% population of Negroes are deficient in glucose 6-phosphate dehydrogenase because these are selected over the normal ones, as these are inhabitants of malaria infested regions. And this glucose 6-phosphate dehydrogenase deficiency is caused by the recessive gene present on the X chromosome. So males are more affected because these have XY sex chromosome, and hence there is only one X chromosome. So one copy of gene is responsible for this disease. Now fourth is industrial melanism. There are two varieties of peppered moth. Scientific name Biston betullaria. One is light colored, which is known as non-melanic variety, and the other is dark colored, known as melanic variety. Now we will discuss about two scenarios. First is before Industrial Revolution. Peppered moths used to rest on tree trunks and rocks. So these tree trunks and rocks were covered with light colored lichens. Hence, against this light colored background, light individuals blended, but dark moths were conspicuous and easy prey for birds. As a result of which, dark colored moths were very rare because light colored moths survive, and most of the dark colored moths were eaten by the birds. Hence, these were not able to reproduce and produce their offsprings. Now, the second scenario. During Industrial Revolution in Europe, that was in 19th century, industrial pollution soot darkened the tree trunk and rocks because these soots covered their surface. Against this darkened background, now dark colored moths blended and light moths were easily visible to the predators. So frequency of dark colored moth in the population started increasing, that is favored reproductively, but most of the light colored moth were preyed, and hence these were not reproductively favored. So what we can say that natural selection, in this case it is predation by birds, edited the population. That is, it resulted in the shift in the composition of Biston betullaria population in industrial regions. That is from light colored moth to dark colored moth. Now let us understand industrial melanism with this figure. This is the earlier situation where there is no industrial revolution. So when the moth rests on the tree trunk, light colored moth easily blend with the color of the tree trunk or the surface, but the dark colored moths are easily visible, so these are eaten by the predators, and hence their percentage of frequency is less in the population. So we can say light colored moth is greater in percentage and dark colored moth are lesser in the percentage. Later during the Industrial Revolution, due to the soots that come from the industries, these cover the surface of the tree trunk. So now light colored moths are conspicuous, but the dark colored moth blends. As a result of which, these are easily eaten by the predators compared to the dark colored moth. Hence now in the population, dark colored moth are more in number of percentage compared to the light colored moth. So with the Industrial Revolution, the population of moth shifted from light colored moth in greater percentage to dark colored moth in greater percentage or frequency in the population. So this is all for today's video. In the next video, we will discuss about types of natural selection. So stay tuned If you like this video, please hit the like button, share it and subscribe my channel. Thank you.